So we'll now start with the second part of our seminar, and our speaker today for this part is Manuja. Manuja is currently conducting a PhD in abyssal ecology at the Marine Sciences Institute in Barcelona and is enrolled as a doctoral student at the University of Barcelona. His PhD project investigates macroecological biodiversity patterns and the community structure of abyssal holothurians in the Clarion-Clipperton zone. He completed his Erasmus Masters through the International Master of Science in the Marine Biological Resources, studying at Ghent University in Belgium, University of Oviedo, Spain, and Sorbonne University in France. And for his master's thesis, he joined the National Oceanography Centre in Southampton, UK, where he explored vertical wall communities in the Galápagos archipelago. His research interests now include abyssal ecology, deep sea ecology, and invertebrate taxonomy. So without further ado, I will hand over to Manuja to share some slides and lead us on his talk.
Okay, so thank you very much for both of you for giving me this opportunity to present my work here. And it's also interesting to hear the first presenter. And I think much of my work is also very related to what Beatrice told and explained in her first presentation. So I'm about to present my work. This work is related to my master thesis project, which I was hosted at the National Oceanographic Centre, Southampton, United Kingdom. And this was 7 or 8 months ago. So my project was supervised by Dr. Loic Oudenheek, Professor Virle, Miss Catherine, and Dr. Kathleen Roberts. But I'm not there at the moment. I'm doing my PhD in Spain. So first, let's look at why it is important to observe these vertical walls. But before we talk about the vertical wall systems, it's important to understand that deep sea is rarely explored. And to give you some statistics, 60% of the world Earth's surface is deep ocean, but only 0.001% is visually explored for the moment. And if you think about that 0.001%, can you imagine what proportion that we have observed on vertical walls in these deep seas? And the other thing is, this project was happened in Galapagos. So Galapagos is known for its endemism. There are a couple of reasons why it has high endemism. I will talk more about the insulation by the water masses within the Galapagos that made it to have high endemism. Also, there's another factor. So these Galapagos Islands are quite young in terms of the geological scale, and they are volcanic in origin. So when you have this volcanic island, they have this high substrate heterogeneity. So when you have the high substrate heterogeneity, you tend to have more niche partitioning. So when you have more niches associated with high heterogeneity, the species they will have species that are more specialized to utilize these very unique niches. So we can expect to have high endemism there as well. And the other thing is, since deep sea is fairly understudied, it is also important to explore these habitats before the climate change can change. So I very recently I read a paper from Hotel et al. I think 2021, I think. So they talk about 7 major shortfalls in the big data, mainly in macroecology. So one of the main shortfalls that they talk about is the Linnaean shortfall. So in the Linnaean shortfall, it is argued that if we don't know about what is there, then we really can't talk about the other aspects of the biodiversity. And what if we don't know what is there, and even though we want to find out, the climate change may wipe out the existing species, so we would never know what would have been there. Okay. And the other thing is that it is also important to understand these deep sea ecosystems in order to develop effective conservation strategies. And you will see why during my presentation, why I talked about these conservation strategies. So in this study, we have three main objectives. Of course, the first one is to record what is there in terms of benthic megafaunal communities. And then we wanted to see the the effect of primary abiotic factors, mainly the site, depth, and the substrate, how these factors shape the community structure. And finally, we also had an additional objective about the vulnerable marine ecosystem indicator species. So, my thesis project was based on the Falkor 2 expedition that happened in 2023, as I remember. No, 2018, as I remember. And the data was already there, but I did the formal analysis, and this data was collected by the ROV SU Bastian. And you can see all these videos that are related to this data collection is on YouTube. So I remember there were some questions from the first presentation that where can we see these videos. So this is a really nice place that any one of you can go here and watch all the videos, and they live streamed everything, so you will get the experience that you are inside the ship. So this is the site map. The red star represent the site locations that I conducted my study: Northern Roca Redonda, and then south of Isabela, then Terrace south of Isabela, and Seamount east of San Cristobal. I originally had the site Cacho de Corals, but we excluded that site because there were no vertical wall reefs we could find from that site. So I followed this framework for the formal analysis. Initially, I identified the timestamps of the ROV video transects. Then I did manual annotations using Beagle 2.0. It's an online annotation platform. And then I created a taxonomics checklist. Finally, I looked into the descriptive statistics, mainly plotting the distributions. And then for univariate statistics, I looked into the alpha diversity components. And then for my multivariate statistics, I used NMDS, hierarchical cluster analysis, Permanova, and simple analysis. And finally, I also looked into the VME indicator species. So this data was collected by an ROV, and it is also important to understand that the traditional ROV data collection is mainly on horizontal surfaces. So if you want to do it on a vertical surface, the setup might be a bit different. And there has been this methodology has not been developed until recently, where one of my supervisors, actually Robert Tettel, in 2023, they developed a methodology how to map these vertical wall communities using ROV and acoustics. So the data we collected, we first wanted to streamline it, because these vertical walls, it's not easy to navigate with the ROV, because the distance from the vertical wall and with the camera can be varied. And with this distance, there will be another variation with the pixel rates. So first, we just wanted to make sure that all the images are good in quality in terms of the pixel rates. And these are some of the frames that I captured from the ROV dives. And you can see here the volcanic origin of this Galapagos archipelago. It gives rises to a unique terrain morphology. On the first image in the left upper corner, you can see the pillow lava structure. So this is where you have this lava formation, and it gradually melts underwater. So you can see this cushion-like structure formation. And when you look into the left bottom one, it is a very near vertical wall with no, very low level of heterogeneity in terms of the terrain curvatures. And there were also other substrate types like like a mix of rocks and sediment, and sometimes dense species aggregates. So I also had another challenge. It is because I need to identify specific frames that are not overlapping. That is something important, because if they were overlapping, then all my analysis would give wrongful inferences. So I did this by manual identification of key features and then manually tracking them one by one for each frames. And as I told you, I did all the annotations within the Beagle 2.0 platform. And for this, I use rectangular shaped boxes. There was a reason why I used these rectangular shaped boxes, because we had an idea in our mind that we will use the same data set for future AI-based analysis. And for the AI-based analysis, it was important to have the boundary area of each annotation as a box. And if you look at the outer margin of this image, you can see it's a bit darker in terms relative to the middle portion of the image. So I wanted to exclude the outer portion of these images from my analysis. And to do that, I placed box in the middle of the image. It is denoted by the red arrow. So everything I annotated was inside this box. But you might also notice that there are some rectangular annotation boxes that are spanning outside of the initial annotation frame. We use this method because if I annotate only a partial image, then the identification of the image within this Beagle 2.0 platform will be really difficult. So in those cases, we annotate the whole organism, irrespective if they are whether they are inside the frame or the outside the frame. The condition was at least a part of that organism should be inside the main boundary layer. And also you can see the two red dots in the middle of the image. Those are the scaling lasers. So uh, between those 2 points, the distance was 10, I think 50, no, 10 centimeters, as I remember. So the important fact here is that we have a scale. So with this scale, we can it can easily be converted to pixel to centimeter scale, and then we can measure the individuals. So that was the important part here, to having having a scale that we can validate. So there was I think Beatrix also talked about this annotation part and the challenges associated with it. So especially in the deep sea, when you don't know what is there, you have a couple of alternative ways how to do this. One way is that you use higher taxonomical hierarchical classification system. Like if you are uncertain about the species, you keep it into higher taxonomy, like species, order, or class. And there's another way that you can also add provisional operational taxonomic units. We call them uh, OTUs. So here you can see for some images, I have assigned certain codes. So instead of putting a name to a species, we use these codes so we can still do the analysis, and there's still room for future taxonomic work to correct these, correctly assign these species names. So maybe even if two species find out to be the same, then we can even merge them. And there's another way. uh, when you are uncertain about this species level classification, you can also go for an open nomenclature system, rather than saying that this is let's say rather than saying that this is Monida Emunida subsolanus, we can say that this is Emunida closely familiar to subsolanus. So the open nomenclature system is also there, and a nice read would be the Horton et al.'s paper, which highlights how to do this in deep sea biology. And this is a very widely applied method when you don't know what is there, but you still want to do research rather than waiting. And there was an estimation, as I can remember, by ocean sensors saying that collection of these deep sea species and actually giving the name by morphological taxonomy, or it can be genetical analysis, can take up to more than 10 years for certain scenarios. So the options are either we do it like this, so you can wait until 10 years to give them actual taxonomic names. So in my research, I had 4 sites, 3 depth categories, 448 image frames, and combined sea flow area of 1,331 square meters, and I did a total number of annotations, 12,458, and everything was done within a period of 5 months, and the actual annotation number is much higher than this. It was around 18,000, but I had to exclude porifera from the analysis, and I just put this image here just to show you that annotation of porifera, the sponges, is practically impossible because their margins were very uh, their margins were overlapping. And I mean, just imagine like trying to annotate a porifera in this frame would take more time unless we find out a way to do subsampling. But during the given time frame for my thesis, it was the best option to put porifera annotations aside. And otherwise, it will also add a bias for the data set just because they are high densities. So now let's talk about the community structure of the epibenthic megafauna in the Galapagos vertical walls. We recorded 123 morphospecies within these vertical wall sections. I mean, compared to the area, this is a really high number. So this is like for every 10 square meters, you find one new morphospecies. And you can already see that where I'm going, because these vertical walls, they are like a small biodiversity hotspot within these uh, deep sea ecosystems. And I, I mean, I explained that I wanted to do descriptive statistics just to have an idea what I am dealing with. But with this image, you can also see that the uh, I mean, at a glance, you can't see anything because it's more or less everywhere. So there's no emerging patterns from these images. So I had to move forward to more complex analysis. To do that, I investigated the alpha diversity components, and this, especially the species richness values for these vertical wall sections. You can see that all the rarefaction extrapolation curves, they reached asymptote. Asymptote means that the curve becomes parallel to the x-axis. So that is where you know that you have sampled much of the species richness in these within these vertical walls sections. Also, we use another approach by Rabon et al. In 2021 paper. Used they used to uh, they used to add all the sites together, and they looked into the cumulative species richness. So in this curve, in the red line, you can see the value I recorded on 123 is I have almost captured the existing biodiversity there. So that is remarkable given that these underwater visual surveys, they say that they usually underestimate the biodiversity, but here in this case, I think we have done a really good job capturing the existing biodiversity from these images. And this analysis was also important because I had the unbiased sampling structure. As you can see, I had three 550 depth transects, then two 400 depth transects, and two 700 depth transects. So when you have a unbiased uh, sampling setup, there are two questions: whether you can see that at what sampling effort are the environmental variables become non-variable. Or you can see, you can ask another question that, have I sampled enough species in order to do any kind of statistical approaches? So here, the first question, the first question to see at what sampling unit when the environmental variables become non-variable is more or less associated with the sample size. When you have a bigger sample size, you can obviously go and answer that question. But in this case, we didn't had that much of a bigger sampling size, but an unbalanced sampling. So we answered the second question. We checked, have we sampled enough species? And with this analysis, we found out that our sampling completeness was well enough to do statistical analysis. And this slide might probably be the most important slide or the finding from this study, because we found out that species turnover was the dominant component in the beta diversity partitioning compared to the species nestedness. So I mean, there are some hypotheses. If you read papers on deep sea macroecology by Rex or maybe McLean, there's a hypothesis saying that in deep sea, this source sink hypothesis, you have this source in the upper layers, and in the bottom layers you have the sinks. So it's like a gradual filtration of the same community. But in these systems, we see that with depth, there might be new species replacing the existing species from the upper layers. So it is something important, because the conservation priorities should go all the way down, because there's species turnover. So it is important in this regard. And the second objective that we wanted to study was the effect of site depth and substratum on this community structure. So in order to do this, I first used PC analysis and NMDS analysis, but you can see that from this analysis, there's nothing much we can infer, because there are no clear emerging patterns from this analysis. So I had to go for another analysis, and here I had two options. Either I go for uh, distance-based redundancy analysis or hierarchical clustering. But instead of going for distance-based redundancy analysis, I went for a hierarchical clustering, because the parameters I have were mostly non-continuous. So when you have mostly non-continuous parameters, it is ideal to use this hierarchical clustering analysis. And here you can see in our cluster analysis, we identified 8 clusters. Here also there's a important point that I need to highlight. If you use like a pure statistical approach to identify these clusters, there are two methods. You can use either shoulder method or silhouette method. Either way, it is like a purely statistical pre-analysis, where you determine what is the cluster number that is statistically significant. But here, instead of going for a statistical significant clustering number, we use the ecological significance of the clusters. So I got the statistical significance of the statistical significance number of clusters were around 6, but we went for 8 clusters just for the ecological significance. And you can see that a limited number of taxa are contributing for the dissimilarity within these clusters. The other important thing that we recorded from this study is that this substrate site and depth, they were acting they had a confounding effect. So we couldn't disentangle any one factor from another that is contributing for these changes. I think it's the same argument that was made by the first presenter. And when you go into these local conditions and very heterogeneous substrate, much of these variables are acting together to give rise to a unique species compositions. But even though there were like uh, confound in nature in these variables. We also observed certain site depth and subset specificities for certain groups. For example, these Echinocipher CF aurelia is the image A I am now talking about. We observed them in really high numbers associated with these sponges or the encrusting biogenic mats. So whenever you have like a bare rock surfaces, we rarely observe them, but whenever the rocky surfaces were covered with the biogenic mat, they are in really high numbers. Also, we recorded Ophiophorus CF aculeata with the dead coral framework, and they were like swarm in these dead coral frameworks. And there's another interesting observation we noticed was in the image D. It's the Monida species identical for number one, and they were like in high densities, but they were inside these little cavities. So it is also not noticeable to highlight that the terrain morphology is also playing a crucial role in terms of species densities within these vertical wall structures. And we also observed that the Metrophora oculata, the scleractinian structure forming coral, was only confined to 400 meters to 550 depth transects. Below that, we couldn't observe any Metrophora oculata coral species, and it's also overlapped with the observations by Salinas de Leon et al. In 2020 in the Galapagos archipelago. So this is the enlarged image. So you can see that these small scott lobsters hiding within these small cavities. And another feature that we observed is having these keystone structures or hyperlocal habitats. As you can see, in some terrain morphologies, they give rise to like high densities of species associations compared to the other areas of the vertical walls. So the argument here is that in sometimes even within these vertical wall systems, there can be favorable microhabitats or niche partitioning that give rise to high densities of species, as well as there can be hydrodynamic conditions that enhance food retention. So this can be the factors what give rise to these kind of species associations. Now let's talk about the vulnerable marine ecosystem indicator species that we recorded during this study. So we recorded 32 VME indicator species, and all these species are listed here. And it is also interesting that different transects, they had different variations of these variations of the VME percentage that observations. So in certain transects, we observe a high number of like 95% or 94% of VME indicator species, while certain transects, they were only like 50% or 45% of the VME. And it is also important to highlight that we had to use three sources from, I think one is from the FAO and one is from this Salinas de Leon et al. Paper that is also from the Galapagos, and another publication that is for, I think, Atlantic Ocean, because within the Galapagos region, to describe these vulnerable marine ecosystem species, we were having a hard time based on the terminologies. And I also remember that the first speaker highlighted that the need of standardization of vulnerable marine ecosystem species and their frameworks. So it is also a gap that we also identified when we were doing our analysis. When we talk about the community structure here, as I explained, the site depth and substratum, they were interrelated and they acted as confounding variables. And we also observed similar assemblages across clusters, and they suggest that complex multi-level interactions, including unmeasured environmental or biological factors, are also shaping this benthic community structure. And there are some directions that I would like to point out because the fine scale terrain morphological features like surface to area ratios or surface rugosity index or slope might be able to explain, maybe able to better explain the unexplained variation of the data sets. I mean, in the beginning, we tried to do it by using acoustic 3D mapping, but unfortunately, we couldn't pull that off due to some software limitations. But the next step would be incorporating these variables into the data system and see if the fine scale subset heterogeneity can answer our question of the unexplained variance in these data sets. And importantly, we also observed all three vulnerable marine ecosystem habitat types defined by the ICES 2020 within the Galapagos vertical walls. So we found coral gardens, we found sponge gardens, and sorry, I can't remember the last category, but this highlights that these Galapagos deep vertical wall systems are acting as biodiversity hotspots within the Galapagos archipelago. And it is also important that this is like a first snapshot of the vertical wall systems and what is the diversity there. So if we want to track the climate change impacts along the vertical axis, so this study provides an initial snapshot. And I would also like to highlight that the continuous efforts needed to investigate the same vertical wall communities in order to have a temporal in order to look at the variations along the temporal axis, which might tell us a different story. If the community composition is changing here, maybe we can relate that to the climate change. And this is also important because I mentioned about the conservation strategies, and since the species turnover is the dominant component within these Galapagos vertical wall systems from 300 meters to 700 meters, the conservation strategies should take the vertical axis when planning for the marine protected areas. So this is a map of the marine protected areas uh, of the Galapagos archipelago. But at the moment, the marine reserve is using the horizontal axis for classify the different regions. But with this study, we argue that the vertical axis should also consider, and you may see why. Here, this is a very recent paper. They say that they rediscovered Rhizosphemia wellingtoni from the Mesopoic zone, I think around 60 meters depth. And they thought to be extinct, this coral species. And we found morphologically very similar morphospecies. But I'm not saying that this is the same exact species. This can be another Rhizosphemia species, but I mean, we don't know. So there can be connectivity from mesophytic reefs to the deeper reefs. And this is not just one-time record. We recorded these very specific morphospecies many times within these vertical wall systems. So this also highlights that the conservation measures should also go along the vertical axis. And yeah, for future directions, it's also important to have multidisciplinary approach. Like You incorporate terrain characteristics, then you also use advanced oceanographic modeling, then larval connectivity studies, and robust taxonomy. If you put everything together, then uh, we can have like very accurate taxonomic support to explain what is there and what are the actual changes, rather than using this morphospecies based uh, analysis. So as a summary, this is the first detailed characterization of the epibenthic megafaunal communities within these vertical wall habitats in the Galapagos archipelago. And we recorded a really high number of morphospecies. It is like 123. And we also found that depth, site, and substrate were confounding, and we couldn't explain like one factor explaining the most of the variation in the dataset. So future studies are needed here, especially using these fine scale habitat terrain characteristics. And most importantly, species turnover was dominant factor, in fact, tenfold larger than the nestedness. And we also observed keystone structures, hyperlocal habitats, and vulnerable marine indicator species. So everything here highlights that the vertical wall ecosystems within this Galapagos archipelago are a vital component in the complex deep sea biodiversity. And it is also imperative to conserve these ecosystems before climate change can move through the vertical axis and change these communities. So I would also like to mention my supervisors, Professor Virle, Dr. Loic, Ms. Catherine, and Dr. Kathleen for their enormous support during the master thesis project. And I also like to highlight the International Coral Reef Society for for funding me. I received the graduate research fellowship during this work. And yeah, thank you very much. That's it from my side. If you have further questions, I'm happy to answer.
That was a great talk. Thank you so much, Manuja. Um, please, anyone who has a question, feel free to type in the chat or raise a hand. While there's typing going on in the chat, I might actually ask a question myself, if that's okay. So Manuja, you mentioned that you worked out what percentage of your sites were, or the organisms at your sites were the VME indicators, but obviously sponges themselves can also be quite important VME indicator species. So I was wondering if, although you didn't quantify them, if you maybe noticed any like really abundant sponges at the sites that otherwise had fewer indicator species.
Yeah, I mean, even though I didn't quantify, I annotated most of the sponges, and we also recognized certain sponge grounds. But we didn't go to species like morphospecies level classification in terms of sponges, but they were definitely there. It's just that the abundance is too high, and if you use any sort of annotation software, it is fairly difficult and time consuming in terms of annotating these species. But as in terms of vulnerable marine ecosystem species, they were definitely there, and they were like a valuable component within these Galapagos deep waters. Yeah.
Were they included in the percentages then? I might have just missed that.
Sorry?
Were the sponges included in your
No,
percentages of the
no.
okay,
They were
okay,
not included in the
okay,
percentages. Only the species that I
okay,
annotated and assigned more for species codes.
okay. I wondered if I just missed that, but
Okay.
that's great. Thank you.
I can spell out some of the questions in the chat. So Beatrice is saying congratulations. And you mentioned in your work that the vertical reefs may serve as a biodiversity hotspots shaped by multi-scale ecological dynamics. So which scale do you think have the strongest influence on community structure? Would that be a local habitat complexity, a depth-related gradient, or a broader oceanographic processes?
Yeah, I think more local habitat complexity would be the best way to go here. And I think mainly the fine scale terrain variables would be the best direction to go, because we wanted to see if the site depth or location or in like any of these variables can answer our questions, but for the moment they were confounding. So I don't think going broader would be the answer. Maybe going more into a fine scale would be the answer, because these are vertical walls and they also have very unique hydrodynamic systems. Like their velocities are relatively high, and the nutrition flow is kind of high along the vertical walls. So I would say that adding fine scale terrain variables, subset heterogeneity, maybe 2D and 3D ratios of the substrate might be a good variable here to be looked into.
So, Manuja, how do you define the vertical walls? Do you have some sort of slope threshold or?
I mean, we can look into the slope, but here is like they are not like 90-degree surfaces. They are like near vertical walls. So we really don't have the quantification because we didn't look into the slope. That's that should be the next part with the acoustic 3D acoustic mapping. Unfortunately, we were trying to do that, but we don't have a specific value. At the moment, they were like estimations of the eye from the ROV explorations.
Another question in the chat, question from artists. So, of all of the creatures you encountered in the deep sea by looking at all those images, which one did you find the most fascinating and or beautiful? That's the first question. The second question is, what percentage of the deep sea creature are bioluminescent?
Okay.
And the third question, or do you want to answer the first
No,
two?
you can go ahead.
What, in your opinion, is the best way for Ant-Farm LK to pass along some of the information that was presented today to fellow artists? Because it is a challenge to understand deep sea research in the way we present it academically.
Yeah. So, I mean, the first question is about what is the most amazing creature that I have encountered in deep sea. So, I mean, at the moment I'm doing a PhD on deep sea macroecology, so it's more about the holothurians. I would say that deep sea holothurians are really fascinating, and I had no idea that the diversity at the abyssal depths, I'm talking about 6,000, can be that high. So you normally expect that going deeper would be lifeless, but it's quite opposite. The diversity is getting really high, but the abundances are getting really, really low. So I would say deep sea holothurians, uh, maybe Paniagoniae, that's the species name, but I think for this question you don't need to give specificities. But if you look into deep sea holothurians, I I think you will get fascinated by their morphology. And for the second question, I really don't know what is a general answer about what percentage of deep sea creatures are bioluminescent. Mm, to be honest, I have no idea about that question, no, about the answer for that question. And for the third one, you're asking me, could you share the best way for me to pass along the two topics presented today? Okay, maybe one way to do is like you can either approach the presenters directly, and they might also give you a nice summary of what they have been talking about in terms of in a very diluted way. And I mean, there are different websites, as the first presenter told. But the thing is, I haven't seen much of the website that talk about the deep sea in a very community friendly way. That's a gap I think that we need to address in the broad sense. Mm, maybe the best way at the moment I think is you talk with experts in the deep sea directly and ask them in a very simple way. Can you explain me this in a simple way? So you can build up a nice conversation with them. Maybe that might be one way to address this.
I think it's a
It's
very
like
good
having
answer.
a one-on-one with experts, so
Yeah, I
you
think
can
most of us
put all
just,
your questions there.
are passionate about what we do and we just love
Yeah,
talking about it.
yeah.
I
I
know that
mean,
whenever I meet people, yeah.
yeah, I mean, in general, we tends to be more scientific because, like, even for this presentation, I was expecting more of like a scientific crowd. Yeah, but I mean, for general public, you really don't want to have this analysis, all the analysis here. I mean, you can build a really nice story with images, and that would make more people wanting to explore these deep seas. Yeah.
So Manuja, in an effort to bridge the gap, uh, what would you say the vertical walls are like significant? Like what's so special about them compared to other deep sea ecosystems that are much more studied?
Yeah, that's true. So, I mean, sea mounts are also special, but if you go along the sea mounts, there's a possibility that you can find near vertical wall sections. And even for these dials, I mentioned that the sampling plan was a bit unbalanced. That's because we really don't know when we find the deep vertical wall. It's like explorative dive. When you find a deep see a vertical wall, you put a waypoint and you keep exploring the area. And after the dive, you go back to the waypoints and then you map the whole section. So you just can't create vertical walls. It's just that it happens to be there and you are lucky to find these deep vertical walls. But similar to sea mounts, they have very unique hydrodynamic systems and nutrition systems. So you expect these ecosystems to have much higher biodiversity compared to other areas, especially compared to the abyssal plains.
And would you say they are more challenging to study? Is it more
Yeah,
dangerous
definitely
for
because
the submarine,
sorry.
or is it more dangerous for the submarine to go there, or like what makes the study of them harder?
Yeah, yeah. So I mean, one point is that the maneuverability of the ROV is a bit tricky. Sometimes you might have a really nice vertical wall, but sometimes there might be like a canyon-like space you have to go in between in order to map these vertical walls. Sometimes you have these overhangs. Underneath you have the vertical wall. So even though you want to map that section, the ROV operator might say, no, I will not go there, risking the whole operation. So that's one limitation. The other thing is the technical challenge. For this specific study, I mean, I was not on board on the ship, but my supervisor and the team, they had to uh, fix the echo sounder in a very unique way. So in a normal survey, you do the sonar like this, right? Because you have the seafloor and you do the sonar like this. But when you have the vertical wall, you fix the sonar like this and it will do something like this. So the whole analysis system you have, I mean, in these kind of systems you have your raw pitch yaw, so all like it's like x y z axis and everything will change. Another different story. Yeah, and when you have a horizontal system, you know the depth from the surface. That's how you get the measurements. But when you have a vertical wall, you don't have a sea surface because it's vertical from the ROV. So you have to create an artificial sea surface away from the ROV. So that's how they get the measurement right. those are the challenges I'd see
Mhm.
in terms of mapping these vertical wall sections.
It looks like most methods have not really been designed
Yeah,
for
actually, the
this.
methods are there because in 2023, one of my supervisors, they developed the method and they tested that method here. And yeah, the method is there. It's just that it's a bit difficult compared to the traditional horizontal mapping because you need to do post cleaning, post orientations, and a lot of statistical analysis.
Maybe we can have one last question before saying goodbye. Beatrice is asking in the chat, how do the communities associated with vertical reefs in the Galapagos compare to those found on deep sea habitats like seamounts and soft sediments?
Let me read the question again. What do you mean by the associated? Can you maybe give more context for that question?
Hi, I can.
Yeah,
Yeah, so you found specific species or organisms, right, in the
yeah.
Galapagos. So how are they compared, the community itself, the community structure, how is compared to those found in different habitat, deep sea habitats? Have you compared that within your work
No,
or
I
in
mean,
other vertical reefs?
no, no. For the moment, we didn't want to compare our work with another deep sea vertical wall. There are some work done in Greenland, as I remember, but it's like a separate ocean there. And the other reason is at the moment I am writing the manuscript for this specific study.
Okay.
So yeah, maybe in future that might be interesting to compare a couple of similar terrain morphologies in terms of this vertical axis, but there's not that much of studies specifically on these vertical wall sections. And if you want to compare like two sites in very different oceans, different depths, that would add a lot of complexity at the moment. So yeah, that is the state for at the moment for this specific study.
Okay. Okay. You can always compare, no in a quantitative way, like making analysis, because obviously different sites are going to show different results. But you can also, you can always compare what is happening with this specific ecosystem that you are analyzing versus other ecosystem. This
Yeah,
give you different answers about different topics.
yeah, yeah. That might be an option. Yeah, but for the moment, yeah, just I'm just writing the manuscript. But yeah, thank you for that insight. It might be a nice
Excellent work.
direction. Yeah.
Excellent work. Congratulations.
Lovely. Thank you so much, Manuja, for your talk and for answering the questions. Can we all give Manuja just another round of applause? And with that, I believe we'll end the seminar for today. So before everyone heads off, I'll just remind you all that our next seminar will be on August the 25th. This will be slightly earlier. It will be at 9 a.m. UTC time. And this topic will be about deep sea sharks. So we will send around a reminder closer to the time, but feel free to put that in your calendars now if that's a topic that you're interested in. Anything else, Maeva?
No, that's right. Thank you, everyone who joined for this opening seminar of the series. And we're hoping to see many of you again in August. And thank you again to our two speakers, to Beatrice and Manuja, for your talks today. Thank you.